So Belantamab Mafodotin, or Belamaf, is an antibody drug conjugate that targets the B-cell maturation antigen. This is very unique and the first in kind in treatment with myeloma. Belamaf works both by the immune-dependent and the immune-independent mechanisms to cause myeloma cell death, including usage of the ADCC and ADCP, makes it a very unique agent that not only targets the cell, but delivers a payload and causes immunogenic cell death as well. So in this specific trial, there were several dose schedules that were tried. So it's a slightly complex trial, so I'll walk you through this. There are two parts of the trial. The part A is the part where Belamaf is combined with lenalidomide and dexamethasone and was primarily evaluated for safety and tolerability and efficacy. In the part B portion, which we're alluding to in the current context, Belamaf is combined with bortezomib and dexamethasone for the same endpoints of the safety, tolerability, and overall response rate. So there are 4 cohorts that are enrolled in this specific part B portion. So, which is in the current context, this is the primary focus of interest. So 2 dosing schedules that were evaluated are 2.5 milligrams and 3.4 milligrams. In each of these dosings, the schedule was slightly different. In cohort 1, the 2.5 milligrams is given on a every 3-week schedule, given as a single dose. In the second cohort, the 2.5 milligram per kilogram is split into 2 portions on day 1 and day 8, with an intent to see if we can preserve the efficacy, at the same time decrease the toxicities that we're seeing. The same dosing schedules applied for the 3.4 milligram per kilogram cohorts, which are making them cohort 3 and cohort 4. What we are presenting at ASCO of this year is the 2.5 milligram per kilogram cohort given every 3 weeks as a single dose, where 18 patients are enrolled. Among the 18 patients that were treated in this cohort of 2.5 milligram per kilogram given as a single dose every 3 weeks in combination with standard bortezomib dexamethasone dosing, a total of, among the 18 patients, 78% had an overall response rate, which is patients achieved greater than a partial response. The clinical benefit rate is seen in 4 out of the 5 patients, which is 83% of the patients achieved a clinical benefit rate. More importantly, more than half the patients achieved a great depth of response greater than a VGPR. So how do these results correlate with the currently ongoing studies in the same area? So with any triplet, with the existing triplets that we have in combination with bortezomib and dexamethasone, these results are aligning in terms of not only the overall response rate, but also the depth of response rates with manageable safety profiles. So in the DREAMM-2 trial, which evaluated the Belamaf as a single agent, where heavily pretreated patients where they've received a median of 6 prior lines of therapy, there was significant response rates that were seen. Close to a third of the patients achieved an overall response rate in both the cohorts that were tested at the 2.5 milligram per kilogram dosing as well as the 3.4 milligram per kilogram dosing. The main toxicities that were seen in the DREAMM-2 trial were thrombocytopenia, the corneal events, and the infusion-related reactions, which is an adverse event of interest. So looking at the DREAMM-6 trial, I'll focus on the exact 3 side effects of interest. Infusion-related reactions, which is of an interest whenever you're evaluating a monoclonal antibody administration. That was seen in 3 of the 18 patients that were treated on the DREAMM-6 trial, which is accounting to 17% of these patients. Mind you, these patients did not receive any pre-medications for decreasing the infusion-related reactions. So it is a manageable, small number, and majority of these are grade 1 or 2 infusion-related reactions.